Ah, yes, DNA replication. In this video, we are continuing our molecular biochemistry series. I think it's episode 4. And in the previous episodes, we talked about the structure of DNA and how we build the nucleotides, how we synthesize the nucleotides of DNA. But now we need to add those nucleotides to the DNA. We can't just make them and then let them freely float around within the cytoplasm or nucleus of the cell. We need to attach them and create new strands of DNA so that that DNA can be part of a new cell after undergoing mitosis or meiosis, or in other words, cell division, because our cells are constantly dividing. And this process of DNA replication begins with a protein known as a chaperone. Now, if you remember that our DNA is tightly linked together through linkers that coil around histones and make up the chromosome. Well, we need to relax the DNA that's linked together so that we can unwind it and then open it up or slice it open for replication to take place. So the chaperone relaxes the DNA, and then topoisomerase, a enzyme, enters the mix and uncoils or unwinds the condensed DNA. And lastly, an enzyme known as helicase does the slicing, also known as the replication fork, will take place. And just a fun bit of knowledge from a medical point of view, there's a syndrome known as Bloom syndrome where a BLM gene is absent that codes for helicase. So helicase becomes deficient and replication fork can't take place. So DNA replication can't take place and the S phase is arrested. And this can cause a number of different symptoms. Anyways, so we have our DNA strand opened up so that we may add a leading strand and a lagging strand so that we have two new DNA helices. And this all begins at the origin of replication, which is always the 3' end of the DNA. And this could be in the middle of the DNA. It doesn't have to take place at the ends. It can be at the middle of the DNA. It could be anywhere within the strand of DNA. And it operates at the 3' end so that the leading strand will go 5' to 3' due to the operation of DNA polymerase, another enzyme. And this guy adds the nucleotides during DNA replication. So remember, we synthesized it, whether through de novo synthesis or through the salvage pathway, and DNA polymerase adds them to the DNA strand that has been forked. And this is a semi-conservative approach, because we have one strand that is part of the original DNA helix and a new strand that is going to allow for a new double helix to form. Additionally, the lagging strand, which is still operating 5' to 3', because that's what DNA polymerase must do, since it is moving away from the replication fork, DNA can only be replicated in a fragmented manner due to this fact. So there are multiple points of origins, I should say, of replication. And it takes place in a fragmented manner where multiple DNA polymerases enter the mix and slowly move 5' to 3', adding nucleotides. These fragments are known as Okazaki fragments. And so this is the main idea of DNA replication. And you can see that there's a lot of reactions taking place, a lot of moving pieces. So it's never perfect. And it's easy for replication to not go as planned or as intended or as how evolution would like it to. And so you can get a lot of different diseases, or the cell will just die. And that's why we have so many cells. And so with that, I will end this episode, and I will see you in the next one.